Today is the last session for this chapter, that is molecular basis of inheritance. Okay, so let's focus on this. Now today is the last topic, or we can say the concept from this chapter. So we'll try to learn, because today we have only two concept, and that two important concept is, one is a genome human genome project, and the another one is a DNA fingerprinting. So here it is totally two different project, I mean to say concept. One is from Human Genome Project up to here, and the another one is a DNA fingerprinting. We will also talk little bit about the genomes. So total number of concept for the today's session is total 3 actually. First one, that is called the Human Genome Project. In short form it is called HGP. Then the another concept we are going to learn today, that is DNA fingerprinting. And very short introductions, or we can say just a little bit about the genomics, because not much question is going to be asked on these questions. Hardly one question is there, so we will see that. Okay, so total 3 concept is there that we will learn in the today's sessions. So let's start with the first one, that is Human Genome Project. But before we are going to start this, you know, as the rule that we have to first recall that what we studied the previous session. So who is going to tell me about that, what we have learned in the previous session? Yes, anyone who remembered, then please tell me what we studied in the last session. As we have number of students over here, so I just want to know that how many of you, okay, gene regulation, okay, and responded gene regulation, okay. What about others? And also lac apron, okay. We studied lac apron, then TRP apron also, right? Okay. So basically, the meaning of gene regulation, that means of how the gene regulate, that reflect the phenotypic effect of the organisms, right? So how actually it is regulated, that we have already studied. So those which joined late or previous session, they can watch the recording session, because it's already. And those who clearly understood, then no problem. Let's go ahead and try to understand about the Human Genome Project. So yes, let's start with the Human Genome Project. Now, what is this Human Genome Project? Now, yet we have very much clear about that human genetic information stored in the DNA, right? Because of the lot of discoveries, lot of research, now we come to conclude that the human, every we can say individual that has identity, you are not exactly similar to your friend, right? Though you can morphologically similar, but you're not exactly identical to your friends, to your colleagues, to your brother, to your sister. So that means of each individual has an identity that is due to one genetic makeup, right? No two individuals are similar. No two individuals are similar. But when I say no two individuals are similar, that means of except the what we can say monozygotic twins. You know, monozygotic twins, because monozygotic twins are genetically similar, identical. So this is the exceptional. Okay, so each organisms is different from one another, right? Now, due to this genetic makeup, you know what, different differences in the genetic makeup are due to the difference in the nucleotide sequence. Why we are different? Why you and me totally different? Though we are human beings, but we have a different morphological characters. So why we are different? Because of the genetic makeup, and these genetic makeup are due to the difference in the nucleotide sequence of their DNA. So basically the differences is, so the result of different nucleotide sequence at the same place in their DNA, means actually the genetic makeup is nothing but it is because of the different nucleotides sequence in the DNA. Means your sequence is totally different than me, and that's why you are different than me. Clear? So keeping this idea in mind, various scientists, because now human beings is totally different, right? So it was there, so at that ancient times, okay, it was therefore always, we can say, a dream for the scientist of the previous era to map the human genomes, right? So it was their dreams, and later on, due to the advancement in the genetic engineering techniques, all these thoughts, idea made it possible to isolate and clone the DNA piece and determine their nucleotide sequence of this fragment. Because it was in the year of 1990. So before 1990, it is very clearly known that the genetic engineering, what is genetic engineering and how it made possible to isolate the DNA, the piece of DNA, and to determine the nucleotide sequence of the DNA. Because you know that DNA, the structure of DNA is already constructed by the Sanger's method, right? By the Sanger's, right? So because of that, in the year of 1990, so 1990, what happened? A mega project, that is Human Genome Project, was launched. By whom? By the US Department of Energy and the National Institute of Health. These two organizations, they came together and they launched one simple project which named as a Human Genome Project. So what is important in this? You have to remember that Human Genome Project was launched in the year of 1990. Clear? So 1990 is the year when the mega project was launched. Human Genome Project was launched. And also by whom? So it was by the US Department of Energy and the National Institute of Health. These two different organizations of the USA, they came together and they launched this project that is called the mega project or Human Genome Project. Now later on, one more institutes, that is called the Wellcome Trust. Okay, let me bring that. Yes, so the Wellcome Trust joined the project. Means later on, once it was launched, then the third partner came and he joined the project and become a major partner in that project. So total how many partners is there? Total 3 partners, right? One is US Energy Department, then the another one is the National Health, right? US Department, yes or no? National Institute of Health, right? So this is 2 department, and later on the another department, that is name is Wellcome Trust. So this Wellcome Trust, all 3 become the major partner of this Human Genome Project. And after that, some other countries also like to contribute, so they join hand with the with this project. And the name of that famous country is that Japan, France, Germany, China, and some other later on some other countries also join hand, and now it become the mega project. Clear? Later on it become a mega project, but initially it was launched by only two departmental institute from the USA, later on Wellcome Trust, and then followed by another countries will also join hand to hand together, and they make this project, Human Genome Project, as a mega project. Okay, is this clear? Yes. Now later on what happened? Okay, before that, later on what happened, one of a very first scientist who started this sequence human genome is known as, is basically known as a John Craig Venter. Let me write the first scientist who scientist to sequence the human genome that is John Craig Venter. Okay, this is the name of the scientist, the first scientist who sequenced the human genome. Okay, so you have to remember this. John Craig, he was a scientist, or we can say he actually PhD student, PhD scholar, and that's why he started. He was a PhD scholar, and along with that, he was a businessman. So he first time sequenced the human genomes. Now, Human Genome Project was launched on 1st October 1990. Okay, as I told you, it was launched in 1st October 1990. Clear? Now, the project was completed in 2003. This project was completed in 2003. So total how many years spent on this project? It was initiated in 1990, right? And it was completed in 2003. So total 13-year, right? So total it was 13-year project. Now another things, as you can see here, it is called the mega project. As I told you, the mega project. Why I called it as a mega project? Because there is something, mega project means definitely if it is big project, so we can say mega project, right? So how? Because at the time of launching this project, the cost which was estimated near about 9 billion US dollar, means when this project was launched, that time the cost estimated, that is estimated cost means what? That is we can say near about the range, which is not accurate, but initial, while starting any kind of work, we have to make a fund, right? So it was a use cost that was estimated near about 9 billion US dollar, and that's what is called the mega project. Not apart from the cost level, but also a very large number of base pair to be identified and sequenced, because they thought that human genome was something near about 18,000 to 150 lakhs of base pair. Okay, so that was the thoughts, and that's why this was something which is a very large uh, sequence to be identified. Another things that make this project as a mega project is the large number of scientists, technician, and even the supporting staff working for these projects, that means of required for this project, and that's why it is a mega project. Now here large number means what? The number is uncountable, so it's a large number of scientists working, technicians, supporting staffs. Okay, that's why this project was considered as a mega project. Even the storage of data, after construction, the Human Genome Project, constructions of Human Genome Project, storage of data generated, which require near about 3,300 books. Means once you create the genome, you require near about, if you want to print it in the form of books, you need to be print 3,300 books. And each book size is 100 page, 1000 pages. Each book, one books contain 1000 page, and on each page it was 1000 type letters, 1000 typed letters. So this was the assumptions, and that's why this was considered as a mega project. Is this clear? Yes, any doubt in this? Okay, now, so as this was a very mega project, because this huge amount of books, huge amount of pages are required. Each book is 1000 page. So you just consider that how many books, how many pages are required, right? However, high speed computational device, because in 1990 everyone know about the computer, right? So high speed computational device for storage, retrieval and analysis of data made it easier to do the same. Because if we write the Human Genome Project by hand, it required near about 3,300 books. But with the help of this bioinformatics technology, bioinformatics, that because computer is now invented in the 1990s, right, before 1990s. So it was very well known that how to operate the computer. So high speed computational device for storage, retrieval and analysis of data made it easier to do the same. And that's why the science of bioinformatics also developed during this period, which helped this Human Genome Project. Right? And that is completed within 13 years of the continuously analyzing speed of human DNA. Right? So bioinformatics is what? Bioinformatics is a new field in which computer hardware as well as software technologies are developed and used to gather, storage, analyze and disseminate the biological data, image and other informations. So this bioinformatics made this Human Genome Project more easier way to construct and complete this mega project. Is this clear? So that is what the idea, the concept behind this mega project, how it was launched. Is this clear to everyone? Yes. So as you can see, the Human Genome Project was a 13 years project which was completed in 2003, and when it started in the year of 1990. Now, Human Genome Project, what is the goal of Human Genome Project? Or simply, what is the methodology of Human Genome Project? Or we can say the different approach of Human Genome Project. So basically there are two important approach of Human Genome Project were analyzed. That is, first is ESTS. What is ESTS? That is Express Sequence Tag. Okay, the first one is Express Sequence Tag and the sequence annotation. This is two important okay, just a minute. So there are two approach for sequencing the human genome. First is Express Sequence Tag or ETS. Why this ETS? EST, sorry, not ETS, it is EST, to identify all the genes that are expressed as a RNA and sequence that same expression, right? So therefore there is a EST, and that mean of expressed sequence tags. Clear? Another one is sequence annotations. Means what? The whole genome, including both coding as well as non-coding regions, are first sequenced, and later functions are assigned to the different regions. So in Human Genome Project, sequence annotation has been carried out, which involve the following steps. I mean, what are those steps? So let's see the later on the steps which are required for the sequence annotations. Just a minute. So disturbing me. Okay. 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 Now this is clear. Express sequence tag and human genome sequence annotation is clear to everyone. Yes or no? Okay. Now let's move ahead. The goal of Human Genome Project. Now let's focus on what is the goal of Human Genome Project. Now you know that because before launching this project, it was goal was already set, and that goal is to identification of all the approximately 20,000 to 25,000 genes in the human DNA, because in the human DNA it was estimated somewhat near about 1 lakh of gene in the human DNA, but at the time of sequencing it was found that only 20 to 25,000 genes. Now another goal is that to determine the sequence of 3 billion chemical base pair that make up the DNA. Okay, so this is the another sequence. Okay, yes, to determine the sequence of 3 billion chemical base pair that make up the DNA. Okay, now third important, that is it also to store this information in the database. What is mean of database? In the form of computational input, right? So that means of whatever the database, that all are going to be stored in the computer. So it is a storage of database into a computational technology. So that is another goal of this project. This is a third. Now another one, improve the tools for the data analysis. Improving the tools for data analysis. Suppose if you want to identify the protein sequence of a particular human beings, so you can just put a sequence on that particular tools. You can able to identify that what kind of tools you have, right? So within a second you can just analyze your data with the help of this project website, right? So that is to improve the tools for the data analysis. Another one is transfer related technology to other sectors. That means of whatever the technologies that with the help of this Human Genome Project, that can be shared with the other sectors to improve the human lifestyle. That was the main motto of this project. Okay, apart from that there are several other project is also there, like it also address the ELSI. What is ELSI? So it is a ethical, social, okay, it is ethical, social, legal and issues, right? Ethical, legal and social issues that may arise from the project. Means whatever the problems arises, that can be solved with the help of this Human Genome Project. That was main motto. Another one, bioinformatics tools. So close associations of Human Genome Project with the rapid development of new areas in biology, this bioinformatics tools can be utilized. That was the goal of this Human Genome Project. Clear? Okay. Is this clear about the goal? One more I would like to add here. This is only 6. So let me add the one more goal. That is 7th goal, that is sequencing of model organisms. Sequencing of model organism. You know what is model organism? How many of you know about the model organisms? Yes, I wanted to know from you what is model organism. You know Drosophila, fruit flies. This is what model organisms. Bacteria E. coli. E. coli bacteria. You might have heard, right? E. coli bacteria. This is also model organisms, right? And there is a Neurospora crassa. Neurospora crassa is also a model organisms. Then there is a yeast. Yeast is also a model organisms, right? So you have to remember the different model organisms. So actually, what is the goal of this sequencing the model organisms? So basically, non-human organism DNA sequence can lead to understanding of their natural capabilities that can be applied towards solving the challenges in the healthcare, because many bacteria are pathogenic. Yes or no? So sometimes we got a bacterial infections in our body, right? So if this non-human organism DNA sequence that can lead to understanding the natural capabilities that can be applied towards the solving challenges, especially in the healthcare, right? If we know the Drosophila habitats, we can solve the problem of agriculture related, energy productions, then environmental remediations. All these are the pathogenics, but that can be used in a good way for mankind, right? Yes, you know. So many non-model organisms are involved in this model organisms. What are those non-model organisms? Let me bring you a one simple list so that you can able to remember that model organism's name, their base pair, and along with that name of the gene. So these are the name of the organisms which you need to remember. So this is this is what? This is a non-human model organisms. This I can call it as a non-human model organisms. Okay, so the name is E. coli. You know E. coli because we have already studied about the E. coli. Saccharomyces cerevisiae, this is called yeast. Yeast. Then there is Synerohabitis elegans. Drosophila melanogaster, which is called the fruit fly. Then there is Arabidopsis oryza sativa, which is commonly called as a rice, right? All these are the non-human model organisms. Clear? So these model organisms can be used in the field of human healthcare, agriculture, medicines, right? So So if we sequence the base pair, we can able to identify the number of gene present. So you have to remember that in E. coli near about 4.7 billion base pair and the number of gene is about 4,000. Similarly, in Saccharomyces cerevisiae, 12 billions 6,000 number of gene. Then Cinerohabitis elegans, which is having about 97 billion 18,000 number of genes. Then Drosophila 118 million 13,000 gene. Arabidopsis, which is about 130 million 25,000 number of genes. And similarly rice, that is Oryza sativa, which is having 430 millions, near about 32,000 to 50,000 number of genes. So these are the name of the organisms, which is very important that you have to remember. So this is called the non-human model organisms. Yes, hope this is clear to everyone. You have to remember each and every model organisms, non-human model organisms. Is this clear to everyone? Come on, responded fast, then I go for the next. Yes, I want response from everyone. Till now 7 responded. Those who not yet responded, please respond it so I can go ahead. Okay. Well, now let's talk about the methodology, or salient features of Human Genome Project. What is the salient features of Human Genome Project? So very first, human genome contain 3,164.7 million nucleotide base. Means our base is near about very a small, not we can say small, but it is an average. So which is 3,134.7 million nucleotide base pair. And the average gene which consists in this nucleotide base is 3,000 bases. So we have total 3,000. But what scientists assumed at the time of launching this project, it was near about 20 to 25,000 nucleotide bases, but it was just a 3,000 base in the human DNA. Now the total number of gene is estimated at 30,000. Almost all 99.95% nucleotide bases are exactly same in all the people. That means in the human beings, means inside you, inside me, inside your friend, inside your colleague, all having 99.9% gene are nucleotide bases are same, exactly same. Just 0.01%. Clear? Or we can say 0.1% gene that is different, and because that 0.1 gene make different you from the other, your colleague, your friends, right? So the functions are unknown for over 50% of the discovered gene, because whatever the estimated in that, only 50% function is known and remaining 50% function is still unknown. Then what is the role of that gene? Clear? Now the fifth salient feature is less than 2% of the genome code for proteins. Protein, you know that how the protein is constructed in human body, right? So less than 2% what genome they are going to code for the protein, means whatever the gene we have, that is 50%, out of this 50% only less than 2% gene can able to code for protein, right? And there is repeated sequence make up very large portions of the human genome. Means repeated portion means what? Like suppose there is a one genome, so there is a repeated sequence. Suppose this is the genome 1, so this genome is repeated in somewhere like here also, then here is, that means there is a there is a sequence which is repeated on very large portions of the human genome, on each and every portions. Seventh, chromosome 1, this is important pointer that you have to note it down. This is important, point number 7, because one questions from Neet ask, I think so in the year of 2021 or 23, there is one questions based on this particular line, that is chromosome 1 has most gene, which is 2968, and the Y has the fewest number, which is very less, that is 231 gene. So that you have to remember. You know that chromosome 1, chromosome 2, chromosome 3, we have how many chromosome we have? Total 23 pair, and their numbering in a karyotypic, the numbering is chromosome number 1, 2, 3, 4, 5, 6, like that till 23. Yes or no? So out of this, chromosome number 1 has most gene, which is the number is about 2900. That means we just consider near about 3000 gene present on the chromosome number 1. So it is the highest number, and the lowest number of gene present on the human chromosome is a sex chromosome, and that is in the case of Y chromosomes. So Y has a fewest number, which is just 231 gene. Is this clear? Yes. So scientists have identified about 1.4 million locations where the single base DNA differences occurs in the human. That means it was also noticed during the this completion of this project. It was also noticed by the scientists that there was near about 1.4 billion locations. Imagine 1.4, sorry, not billion, it's a million. So 1.4 million locations in that human genome sequence that where the single base DNA differences are found. So this was something, these are the salient features that you have to remember. Clear? Now, okay. So another salient features I would like to tell you that the length of, the total length of the human, the difference of human genomes is very different, very widely, right? So the average gene size is 3000 base pair. As we know that we have the chromosome number 1 having the gene size which is 3000 base pair, 3000 gene pair, right? So beta globulin and insulin genes are less than 1 kilo base pair long, right? That is less than 1 kilo base pair. The largest gene of human body part is that is called the Duchenne muscular dystrophy, which is present on X chromosome. That is the largest number, largest what in the sense of big and small size. I'm talking about the size. Clear? So that is what the name of that gene, the largest gene of human body body, that is called the Duchenne. Yes, it was Duchenne muscular dystrophy. Okay, and where it is present? It is present on X chromosome. On X chromosome. This is what, this was the largest gene of human body part. Largest gene of human body part. Is this clear? Okay. Now, and the most smallest gene. Sorry, let me tell you the number. What is the number of that largest gene of human body part? That is the number is near about 2.4 million. Yes, the number is 2.4 million. It is 2.4 million. Okay, but when you convert it into this million, it become a 2400 kilobase. KB, in short it is called KB, right? Base pair long. Similarly, this is what the largest gene of human body parts. Similarly, there is a smallest gene, smallest gene of, that is the smallest gene of that, that is called the TDF, that is testis determining factors. TDF, it is testis determining factor. What is that testis determining factor? Where it is present? It is present on Y chromosome. Yes, it is present on Y chromosome. And what is the number? So the number is just only 14 base pair. Yes, just only 14 base pair. That is what the number. Is this clear? The largest means large in size and the smallest means smaller in size of gene in the human body. Is this clear? Yes, responded fast. Okay. Another one.
Sir, in SNP, they are saying that in a single base pair, it will be different from the uh, other humans.
Like what is the question? Repeat again.
SNP is a single nucleotide polymorphism. So they're saying that in the entire genome, only one like nucleotide is different, and that is 0.1% different.
Yes, 0.1%.
Okay, sir.
Okay, so this is about the largest and the smallest. Now let's talk about the repeated sequence. The part of DNA, because the salient features of this Human Genome Project, it is mentioned as the repeated sequence, right? So what is that repeated sequence? So let's talk little bit about, because that is also important. So yes, now the repeated sequence. Now what is repeated sequence? I told you in that the goal of the Human Genome Project, in that I have already told you that repeated sequence. Now this repeated sequence of DNA, that is called the satellite DNA. Okay, repeated sequence of DNA, that is called the satellite DNA. Okay, it is called the satellite DNA. Now this satellite DNA is of various type, or we can say there are various type, but we will see the major one. So out of this, one of them is called the moderate repetitive sequence. The first one is moderate repetitive moderate repetitive sequence. Another one that is called the mini satellite sequence. Mini satellite sequence. Then this mini satellite sequence, or another one is micro satellite. Yes, one more is there. Micro satellite sequence. Clear? So what are these different type of sequence? So very first, as I told you, that the part of DNA which contain a repeated sequence. Repeated sequence means, suppose, let me draw the line. Suppose this is a particular line or particular sequence. Okay, and in this sequence there is, this is the repeated sequence which I'm drawing over here. This is one, another one, like this, right? So this is what the repeated sequence. This is the sequence number 1. I can write it as a K. This is K1, K2. So all these are the repeated one, right? So this repeated sequence is what it is called the satellite DNA. So the part of DNA which contain the repeated sequence, then it is called the satellite DNA. Now this satellite DNA classified into 3 types, or there are several types, but we will focus on these 3 types. What is this? The first one is called the moderate repetitive sequence. Moderate repetitive sequence which having near about 150 to 300 base pair long. It was 150 to 300 base pair long. Okay, very first one. And there are several, another one, out of which one is called the mini satellite sequence, which having 11 to 16 base pair long. This is having 11 to 16 base pair long. My pen is visible to all of you? Yes, this is what 11 to 16 base pair long. And this 11 to 16 base pair long, they are referred as a VNT, VNTR. What is VNTRS? It is nothing but, we can call it as a satellite, a kind of, these VNTRs basically use in a DNA fingerprinting. Yes, so we will focus this DNA fingerprinting in the next what we can say concept, means today itself only, in the next concept, and that's why you have to just keep in mind what is this VNTR. So this is a part of mini satellite sequence. Okay, it is a mini satellite sequence. So when you the full form of this VNTR is variable number tandem repeats. Okay, it is the full form number that we will see what is variable number tandem repeats in the DNA fingerprinting. Okay, which is the next concept after this. So mini satellite. Now another one which is called the micro satellite. This micro satellite, that is basically it is found in the genome, and they have 5 to 8 base pair. Okay, it having 5 to 8 base pair long. Okay, so repetitive sequence, mini satellite, and the micro satellite, all these are the part of satellite DNA, different types of satellite DNA. Because in our syllabus only these 3 which is important, and that's why I'm just repeating this 3 one. There are several others also So if you want to learn, you can just Google it. You will get a number of microsatellites and satellite DNA, but these are the major ones. Okay. Now let's move ahead. Okay. Yes. The next is, so now we completed the process, right? Okay. Just 1 minute. Are we discuss about the methodology? Just a minute. Okay. Okay. Let's talk about the methodology. That I think so. Yes, forget. So let's start with the methodology. That means of while constructing the Human Genome Project, what kind of methodology they followed, that we are going to learn here. Yes. So now come to this particular diagram. Now what this diagram represent over here. So basically this diagram, it relate to that there are basically, as we have already studied, that there are basically two methodology, or we can say two approach for the sequencing Human Genome Project. One is referred as express sequence tag, that is called let me write ESTS. The second one, that is called the sequence annotation. Yes or no? This is the two important approach of the Human Genome Project. Clear? So the whole genome, including both coding and non-coding regions, are first sequenced in that Human Genome Project, and later the function are assigned to a different region. So in this human genome, sequence annotation has been carried out with the help of the diagram. You can see the whole DNA of cell is isolated. That means of, you can see here, this is the human beings. Any human, just consider this is human. I'm not writing as a male or female. This is one particular organisms. Okay. So in that particular organism, cell is extracted. So this is a cell, yes or no? So cell is extracted from the organism. Clear? And there is a broken randomly into a fragment, means this cell is going to be crushed and into a fragmented. That means of it is in a part of the. So one it crusted, you can easily get the. So in that out of you will get the chromosomes, right? So this chromosome or these particular fragments, they are inserted into specialized vector, and that vector is called the vectors like BAC and YAC. They are called the bacterial artificial chromosome, and YAC stand for, it is a yeast artificial chromosomes. Why I said yeast and bacterial? Because these are the non-human model organism. Yes or no? Yes. So yes. Now this is total 5 important points that discuss the entire methodology. Okay, so very first, what happened? The first, they have the whole DNA of the cell is isolated and broken randomly into a fragment. Now this fragment is going to introduce into a specialized vector. That vector is called BAC. Means you have a fragment of DNA like this, and this fragment convert into a BAC. BAC is what? It is a bacterial artificial chromosomes. So when you insert this bacterial artificial chromosome, it is going to produce similar kind of clone, right? So the fragments are clone in the suitable host. Who is host? Bacteria and yeast, right? Because you are inserting inside it. So when it inserted, that is going to be make a clone of that inserted part. So what happened? So this is going to make a clone, and the PCR can also be used to cloning or making the copies of DNA fragment, because PCR is another method which is called the polymerase chain reactions. So you can multiply your number of fragment in within a second or within a minute, right? So PCR is also one more kind of, this is artificial method of making the cloning, or we can say it is making the number of copies with the help of PCR, or either with the help of bacteria or yeast. So can also be used for making the, or making clone, or we can say make a copies of DNA fragment. Now these fragments are then later on what? It was sequenced as a annotated DNA sequence, right? So as you can see, the annotated DNA sequence. Then the sequence were then arranged on the base of their overlapping region, and its necessities the generation of overlapping fragments for the sequencing, and that is going to be computer based program were used to align the sequence. So with the help of computer, this all sequence is going to be aligned. So with the help of it is going to be analyzed, and the sequence were then annotated and assigned to a different chromosomes. So this is what the complete methodology, how they created this Human Genome Project. So if you want to, so in the future, if you want to create a genome of any particular organisms, what steps you have to follow? First of all, you have to isolate the cell. That isolated cell is going to be broken down into a fragment. That fragment you're going to insert into a specialized vector, like we can say any vector you can take, but here is specifically taken as a bacterial artificial chromosomes or yeast artificial chromosomes. So you can take a suitable vector, and that when you insert it into that vector is going to multiply the number of the particular fragment, and that fragment are then sequence annotated DNA, and then later on with the help of computer based program it was aligned to the sequence. So this is what you will get the complete genome of the particular organisms, right? This is what the methodology followed by the creator of Human Genome Project. Clear? Is this clear about the methodology? Yes, anyone have a doubt in this? I want response from everyone. Okay, so what we studied about this Human Genome Project, we started with the actual how this project was initiated, right? Then we studied the why this project is called mega project. We also studied about the goal of the project, that what was the goal of this project. We also studied about the methodology, that what kind of methodology they adopted to create this Human Genome Project, like as we just now saw. And we also studied about the salient features of Human Genome Project. Clear? Now let's focus on the application and future challenges of this project. Okay, so hope this Human Genome Project is very clear to all of you. Yes? Once I say yes, no one responding except Ann and Sarjana. What about others? All are sleeping? Come on, respond it fast. All are sleeping. Okay. Now let's talk about the application and future challenges of this Human Genome Project. So very first application is molecular interaction. Now, what is this molecular interaction? So let's focus on and try to understand the molecular interaction. So molecular interaction means what? All the gene in the genome can be studied to understand how they work together in a interconnected network. Means how one organism is interconnected with the other organisms. So this, to understand this behavior, we have to go for molecular interactions. Then only it is possible to establish a connection, communication between the two organisms. So basically it is a future application is molecular interactions. Another one, disorders. So knowledge about the effect of DNA variations among the individual may help in a better diagnosis. Suppose if because everything is a in the human body is a DNA. If any a person having some kind of disabilities or some kind of disorders, so if a doctor know that disorder because of the DNA label, so they can treat and hopefully prevent from the many disorders that affect through the human beings, right? So that means of the another, and the second application is disorders, right? Why disorders? Because if you know the particular DNA is defective, then you can treat and remove that DNA. Because we are, so which DNA is defective, that we can able to analyze with the help of this Human Genome Project. Clear? Yes. So if anyone have any doctor, if you if know they have knowledge about their effect of the DNA variations among the individual, definitely they may help in a better diagnosis, treatment, and may hopefully they prevent many disorders that affect the human beings. Clear? Another one is a non-human organisms. Now you know very well what is non-human organism. So by understanding the DNA sequence of non-human organisms, what are the non-human organisms? I've told you that model organisms, right? So by understanding that DNA sequence of non-human organisms, informations about their natural capabilities can be applied toward the solving challenges in the healthcare. Like suppose there is a one disease which cannot be curable. Like we can say the AIDS is a kind of disease which is not curable. But if we know the understanding of the genome DNA sequence of every of non human organisms, so we can able to analyze that how we can solve this kind of, how we can treat this kind of Like, so that means of you can able to solve the challenges in the healthcare, in the agriculture, even in the energy productions, right? And in the environmental remediations. So all this can possible if you understand the DNA sequence of non-organisms. Now, environmental remediation means what? There are so many bacteria available in the atmosphere that are environmental remediate bacteria, right, which is called the xenobiotics bacteria. So that bacteria can be clean the pond water if the pond is full of lots of heavy metals in that pond. So that bacteria, when you introduce in that particular pond, they will clean the entire heavy metals from that pond. So that can be possible if you understand the DNA sequence of non-human organisms. So we can utilize that DNA sequencing in the area of healthcare, agriculture, energy productions, and the environmental remediations. Is this clear? So in this non-human organisms, the different model organisms I have told you earlier, total 6 model organisms, non-human model organisms, 6 or 5, 6 or 5. Tell me the number, exact number. It is 6 or 5. Tell me fast. What is the number? 6 or 5? 6. Okay. So anyone can tell me the name of that all 6? Don't write it. You can unmute and speak. Non-human model organism. Tell me fast. No one. No one.
Sir, we have E. coli, that is Escherichia coli, then Saccharomyces
Yes.
cerevisiae, which is
Service.
yeast, then
Yes.
Drosophila melanogaster, fruit fly, then Orsia sativa, which is
Yes.
rice,
Yes.
and Cenohaptitis elangus.
Cenero habititis
Cenohaptitis elangus, then
elegans.
Arabidopsis.
Yes. So total 6 non-human model organisms, and this can be sequenced. This has been sequenced, not can be. It is already sequenced to understand the connection between the two organisms, because all these organisms are used as a model organism in the recent, and that's why this has been already sequenced. Clear? So this is the third application, or we can say the future challenges. Yes. So we completed the Human Genome Project. Now I want to give you break for 5 minutes. Yes. See, Sajna smiled. Why? You want to break?
Yes, sir. I actually wanted a break, sir.
Okay, okay, no problem. So whenever see anyone want to break, then at least, but not always. Sometimes, like suppose in our 2 hours and 30 minute session, if you want to break in between, so definitely you can say. So I can stop within for 5 to 10 minutes for that not 5 to 10 minutes, but at least for 5 minutes for the break. Okay. So yes, I'm going to give you break. Now it is 8:15. Now you're all present at 8:20. So 5 minutes break you can take, and then we will start the next concept, that is called the DNA fingerprinting. Clear? So in the next session we will talk, next session means in the next concept we will talk about the DNA fingerprinting. So you all can take a break for just a little break and then come back again. We'll start at sharp, we'll start at 8:20 because it's 8:15, right? So you all can take a break. Yes, Sajana, you can go now. Hello. Yes, all of you present. Yes, on your front camera. Now break is over. Sasidharan, are you there? Sasidharan, are you there? Yes, just respond in the chat box. How many of you are present? Please respond it in the chat box. Yes, Prasanti. Okay, Prasanti present. Bhuwana Sri present. Okay, Sarjana present. Okay. So only 3 students present. Ragul, are you there? Ragul, Sasidharan, Gopinath, and come on, respond it. Yes, come on, respond it. How many of you are present right now? Come on, respond it so I can start this concept. Prasanti, Bhuwana, and Sarjana present. I just wanted to. Okay, Anne is also present. Ragul, are you there? Ragul, Sasidharan, Gopinath. Ragul, Sasidharan, Gopinath. Please respond it. Okay, so well, now let's start and focus on this DNA fingerprinting, which is very important. Now let's go a little bit about the history, that how the DNA fingerprinting started, or what is exact mean of DNA fingerprinting. So the study of finger, palm, or we can say the sole print, is called the dermatographics, because this kind of finger print or palm or sole print is generally determined by the different ancient. It is already mentioned in the ancient history, right? So that is what, when you study the finger, palm, and sole print, this is called dermatography. Yes, let me write here. It is called the dermatography. Sorry, it is called dermatography. Dermatography, or sorry, graphics. Yes, it is called dermatographics. Okay, that means of when you study fingerprint or palm print, or we can say the sole print, it is called dermatographics. Now this kind of it has been always a subject of human interest since the ancient times, when we started used to hunt for the food with the help of animals footprint, right? So the science of fingerprinting was first, it is used by the Sir William Herschel. So you can see it was first used by the William Herschel as a method of identification in the year of 1858, not 19, it is 1858. In India, the science of fingerprinting was discovered by chance during a murder investigations in the Jalpaiguri in the year of 1897. Jalpaiguri, where is Jalpaiguri? Anyone know Jalpaiguri is in which state? Come on, tell me. If you know, then say no. Jalpaiguri is in Bengal. You know Bengal? Bengal. Are you not all responding? What happened to all? Is there any problem? Then no one responding? Okay. Okay, so it is in Bengal. So it was Alec Jeffrey. The name of the scientist is Alec Jeffrey. This is slightly mistake. This is not a William Herschel. This is Alec Jeffrey. Jeffrey. Okay. So Sir Alec Jeffrey, who in the year of 1984 invented the DNA fingerprinting technique. So that means a technique is invented by this Alec Jeffrey. But DNA fingerprinting started in the year of 1897. As you can see, it is already started. The science of fingerprints was discovered by chance during the murder investigation in Jalpaiguri in the year of 1897. But actual DNA fingerprinting, it was started by the Alec Jeffrey. I mean, it is invented by Alec Jeffrey, the technique, and that is in the United Kingdom. Clear? Now, in India especially, Dr. V. K. Kashyap and Dr. Lalji Singh, you can see the Lalji Singh, started the DNA fingerprinting technology in India at CCMB. What is a CCMB? It is a Center for Cell and Molecular Biology, which is in Hyderabad. Okay, so it is also this man Lalji Singh is also called the Indian father of DNA fingerprinting. It is also, this man is also referred as a Indian father of DNA fingerprinting. Okay, that is Lalji Singh. Okay, so Alec Jeffrey is a father of DNA fingerprinting and Lalji Singh is the father of Indian fingerprinting. Okay, so don't get confused. Alec Jeffrey is father of DNA fingerprinting. DNA fingerprinting, and Lalji Singh, Dr. Lalji Singh, is father of Indian fingerprinting. Father of Indian fingerprinting. Clear? So this two you have to remember, that is Alec Jeffrey and Dr. Lalji Singh. Now, what is the principle behind this DNA fingerprinting? So yes, the principle behind this DNA fingerprinting, or simply what is DNA fingerprinting, it is nothing but it is the DNA fingerprinting is also called the DNA typing or DNA profiling. Let me tell you very much clear that what is DNA fingerprinting. This DNA fingerprinting is also called, it is a DNA typing. DNA typing, or you can call it as a DNA profiling. Okay, so DNA fingerprinting is either it can be called it as a DNA typing or DNA profiling. Now it is a technique. This DNA fingerprinting is a kind of technique that determines the nucleotide sequence of certain areas of DNA. It, what is the main aim of this technology? It going to determining the nucleotide sequence of some specific region of DNA which are unique to each human beings. That is what called DNA fingerprinting. Clear? So this is the definition that what is DNA fingerprinting. DNA fingerprinting is a technique for determining the nucleotide sequence of some specific region of DNA which are unique to each human beings, like which make difference between the two different organisms, which make a difference between you and me. That is what called the DNA finger. So with the help of the technique we can able to determine that nucleotide sequence. Clear? Now this specific region, now as I just told you the specific region over here, right? So this specific region, this specific region, so these specific regions are called the repetitive DNA. And you know what I said about the repetitive DNA in the Human Genome Project? It is called the satellite DNA. Yes or no? Yes or no? Right? So these repeated DNA sequence are separated as a satellite DNA from a bulk genome as a different peak during the density gradient centrifugations. So what is this density gradient centrifugations? It is a kind of techniques through you can able to centrifuge and sorted the DNA according to their size. So on the base of that size you can able to sorted the DNA from the largest fragment to the smallest fragment, and in this way you can separate each and every individual DNA segment. So that is possible with the help of density gradient centrifugations. Now, the satellite DNA, why it is need to segregate the satellite DNA? Because satellite DNA can be classified as a microsatellite. I have told you about the microsatellite and mini satellite also, right? So they are classified as a microsatellite or mini satellite depending on the base compositions. Means whether they having AT rich region or GC rich region. The length of segment and the number of tandemly repetitive units, that is called VNTRS. And what is the full form? Variable numbers of tandem repeat sequence. Clear? Is this clear? Okay, so that is VNTRS. Now this repetitive sequence shows a high degree of polymorphism. Now what is polymorphisms? So you know that the mutations in the non-coding sequence have piled up with the time and form the basis of DNA. So these polymorphisms, that is variations at the genetic label, arises due to the mutations. So this is what the polymorphism. So DNA polymorphism is the basis of genetic mapping of human genome as well as the DNA fingerprinting. So you can see in this diagram a short nucleotide repeats in the DNA, short tandem repeats or nucleotide repeats in particular DNA in particular DNA are very specific in each individual and vary in number from person to person, right? So these are called the variable number of tandem repeats, or as I told you, that is VNTR. This is slightly different from person to person, VNTR, right? So this VNTR, these are called the mini satellites, and each individual inherit these repeated from his or parents, which are used as a genetic marker in the personal identity test, right? So basically this particular VNTRS, or we can say the mini satellite, basically it is used to identify the person's identity, like whether this particular child belongs to first parent or the second parent. That means how to find out their genetic constituents, this VNTR or mini satellites is used to identify the particular DNA, right? So that indicate, so half of the VNTR alleles of child resemble that of the mothers. Means if a particular child having VNTRS, so that VNTRS is 50% from father and 50% from mothers. Clear? So it can be easily concluded that this particular child belongs to these parents, right? So this is what, this is nothing but this is a variable number tandem repeats, VNTR, which is also called the mini satellite. Now let's try to learn. This is DNA fingerprinting, that what are the techniques and how it is used. Now, what are the steps required for DNA fingerprinting? So basically, if you want to perform a DNA fingerprinting, what are the basic things you require and how you will perform this DNA fingerprintings? So there are several steps. So very first step is cell sample. Means any kind of sample if you have, like suppose any plant cell, any animal cell, any cell if you have, you can isolate the DNA. That means of, you know, the DNA present in the plant cell or animal cell or any kind of organisms. Yes or no? Yes or no? Yes. So that means of, in the case of plant, you can isolate the cell, and inside that cell there is a DNA. So you have to extract the DNA from that cell by what technique? By using the gradient centrifugation techniques. So you can easily isolate the DNA. Now DNA is what? It is a thread-like filament, right? So you have to break that fragment into small pieces, into a fragment. So how you will break? So with the help of DNA enzymes, that is called the restrictions endonuclease. Okay. So first DNA sample, cell sample, then there is a DNA extractions, and this extracted DNA with the help of enzymes called restrictions enzymes. That restriction enzyme is called the restrictions endonuclease. Restrictions endonuclease. Let me write here. It is restriction endonuclease enzymes. Okay. So what is this restrictions endonuclease enzyme? It is a kind of scissors who going to cut the long thread into a particular segment. So you can say this is the complete long segment over here, and this is going to be cut into a small fragment. So this is one fragment, this is another fragment, this is another fragment. So with the help of enzymes, you have to remember that is restrictions endonuclease enzymes. Okay, is this clear about the step 1, 2 and 3? This is step 1, this is 2 and this is 3. Any doubt in this? Step 1, 2, 3. Okay, now what is the next step? The next step is the fragments are separated according to the gel electrophoresis. Now what is this gel electrophoresis? This is a kind of gel electrophoresis. Okay, and when you load your DNA, this is what the fragment of DNA, this is a fragmented DNA. Yes or no? So this fragmented DNA, because you cannot handle it with the forceps, so it is in the form of liquid. So with the help of micropipette, you have to just load this DNA on this gel electrophoresis sheet. This is the gel electrophoresis sheet. So you have to just load it over here. Here is a well and you have to load it on this well. So what happens after certain times, that DNA is going to separate according to their size. So if there is a larger DNA, it is at the base. If there is smaller DNA, it reaches to the apex. Why it is so far? Because those DNA which having a larger molecular weight, it will go slowly, and those having smaller molecular weight, they go fast, and according to their size they are going to be separated. So here, which is present at the top one, this is the smallest DNA fragment, this is the largest DNA fragment. So based on that, you can able to separate the DNA fragment with the help of gel electrophoresis. This is a gel electrophoresis. Is this clear? The step number 4. Yes or no? Okay. Now after that, what is the next? Now this electrophoresis, this gel is going to be transferred in the another membrane, and that is called the nitrocellulase membrane. This is a nitrocellulase membrane. Let me write here. This is called the nitrocellulase membrane. This is nitrocellulase membrane, or it is also called as a nylon membrane. Nylon membrane. Okay, so you can see here, this is a one sheet which is placed over here. So there is a kind of gel, like this is a gel and lot of DNA fragment present over here. You have to just keep one nitrocellulase membrane over it like this. So what happened? All the DNA fragment is going to be loaded on the nitrocellulase membrane. Now binding of radioactive. Now then what is the next one? Now you have to add the radioactively DNA probe. After this, this is the 5th step. Now in the 6th step, you have to add the specific probe or DNA probe on the 6th stage of the DNA fragments. And after loading the probe, now what is the next? The next is the fragment of particular size having V and TRS are multiplied with PCR, that is polymerase chain reaction technique, and they are treated with alkaline chemicals to split them into a single stranded DNA. Means initially it was a double, but when with the help of VNTR, it is going to be separated and it become a single stranded DNA. Now the separated fragment of the single stranded DNA are transferred into a nitrocellulose membrane, as I told you, and then there is a radioactive DNA probe which you need to add inside. So some of them will bind a single stranded VNTR, and the method of hybridizations of DNA, that which probe is called the southern blotting. So here you can see, this is what the method when you using the blotting paper, or we can say nylon paper on this, it is called southern blotting. So southern blotting, SOU, it is called southern blotting. This southern blotting is especially for DNA. Okay, so this technique is called southern blotting. Now after southern blotting, Now you can see the nylon membrane is washed to remove the extra probe, like this is the nylon membrane. Now you have to wash this nylon membrane. So if there is an extra probe, that can be easily washed out, and there is a required probes which are present on this nitrocellulose membrane. Now this nitrocellulose membrane, an X-ray film is exposed on this nylon membrane to mark the place where the radioactive DNA probe have been bound to the DNA fragment, and these fragments are marked as a dark band. When X-ray film is developed, this is known as autoradiography, because it is based on auto. So it is, initially it was X-ray, so X-ray film used to detect the radioactive, but now it is used by autoradiography. Autoradiography. Clear? So this is autoradiography. So the band, dark band on the X-ray film represent the DNA fingerprinting. You can see here this band. You can see. Can you able to see this band on this? So this is what this band represent, the DNA fingerprints, or it is called the DNA profiling. Is this clear? Yes. Now, see, I would like to give you one important flow sheet that will help you to understand. See here, suppose now in the previous case we studied about the plant cell. Now this is the animal cell. Like suppose you have a blood sample in any crime place, you have collected a blood. You don't know that who made this crime, right? So you have collected the blood sample. Now how to identify the suspect? So you have to first collect the blood sample, then you have to isolate the DNA. So DNA is extracted from the blood sample, then it is going to be cut into a fragment by the restriction enzymes. What is the name of that enzyme? Restriction enzymes? Restrictions endonuclease enzymes, right? So you have to cut it with the enzymes, and after cutting, then what is the next? You have to the DNA fragment are then separated onto a bands during the electrophoresis on the agarose gel. So this is the agarose gel on the so on this agarose gel you can able to separate the DNA according to their size. Once it is separated, the next step is the DNA band pattern in the gel is transferred to a nylon membrane. You can see it is transferred to the nylon membrane. This is the upper one, which is nylon membrane. Okay, by the technique that is called the southern blotting technique. So how you will transfer the DNA with the nylon membrane? This technique is called the southern blotting. Now after southern blotting, what is the next step? Now the next step is that the radioactive DNA probe is prepared, and that you have to insert on this the DNA probe band to the specific DNA sequence on the membrane. So on this membrane you have to add the probe. Okay, so this is the sequence is very important. You have to remember each and every sequence. This is the first sequence, second, third, fourth. Fourth means this one, fourth, then fifth, this is the sixth to prepare, and seventh is to add. So directly we can say seventh can be also be the add, to add the probe DNA to on this. Then excess DNA was, suppose there can be excess DNA. So that excess DNA you can wash off with and then you can see there is a, at this stage the radioactive probe is bound to the DNA pattern on the membrane, and then the X-ray film is placed next to the membrane to detect the radioactive pattern. So here you can see you will get this kind of pattern, and this pattern represent the DNA fingerprinting. So this is the pattern which represent the DNA fingerprints. So you can easily able to identify the suspect from this type of technique, which is called the DNA fingerprinting. Is this clear about the technique of plant sample as well as animal sample? Yes. Anyone have a doubt? Okay. Now let's talk about the application of DNA fingerprinting. So basically where this DNA fingerprinting is used. So first it is nothing but it is called the individuality. So individuality means what? Like a pose, like no skin fingerprinting. DNA fingerprint can help to identify one human being from the another human beings. But this is exceptions in a monozygotic twins. You know, monozygotic means both the person having the same morphological character, means their fingerprinting are also similar, right? So to identify a specific person or individuals, then this DNA fingerprinting is applicable. Like suppose uh, in ancient times, farmers basically they are illiterate. Okay, so they generally sell their property by using a thumb. Like they have to sign, but instead of sign, they generally use for thumb process, right? So, so if suppose there is any false thumb, how to identify? So with the help of this technique that is called the DNA fingerprinting, we can identify a one human being from the another. So whether this person is belongs to same or that different, that we can able to identify. So very first it is to identify the individuality. Okay. Now the another one, that is paternity or maternity dispute. Suppose there is one child and two parents claiming that this child belongs to them. Okay, so there is a dispute. Now the matter is in the court. So court decided to do a DNA fingerprinting. So how they identify that this child belongs to this parent, right? It is with the help of DNA fingerprinting. So DNA fingerprinting can identify the real genetic mother and father and the offsprings, because VNTR, VNTR is mini satellite, 50% from parent, 50% from mothers, right? So with the help of this technique, they can easily identify the paternity or maternity of the particular child. Clear? So this is the application of DNA. Another one is a human lineage. Now what is human lineage? So in the case of human lineage, basically we can say the DNA from the various probabilities being studied to find out the human lineage. Like suppose if you want to find out what is the origin of human beings, so to identify the origin of human beings, we can do DNA fingerprintings with the help of so chimpanzee, then a monkey, chimpanzee, and then there is are various of human beings lineage. So with the help of that you can identify and make a hologram of complete human beings. So here also DNA fingerprinting is important. Another one, that is heredity disease. Like if any parent, their grandfather or mother having some kind of heredity disease, so with the help of DNA fingerprinting, this technique is being used to identify the genes which is connected with the hereditary disease, which gene is responsible such kind of disease. So that can be possible with the DNA fingerprinting. Even in the forensic science, like suppose like here in this case, you see here, this is a very beautiful diagram. Okay. Now here, forensic science. DNA fingerprinting is very useful in the detection of crime and legal pursuits. suppose the DNA fingerprinting has been proved that suppose here is a victim, this is a victim, and there is a crime scene. Now there is a 3 suspects, 1 suspect number 2, suspect number 3. Okay, so the sample collected from the victim, okay, and from the suspect, and we can analyze that with the help of DNA fingerprinting. We can analyze the band, so you can see which band is more closer. So you can just, with the help of this band pattern, you can identify this is the crime scene. Okay, so at the crime scene there is a blood sample, this is the pattern of fingerprints. So you have to match this fingerprint is more similar to whom. So you can see this is matching to this, than this, but here there is a no band, but here is a band, means this is not a murderer. So now we have to, now you can see this is also matching with this, but not this, so this is not a murderer. So he is, suspect number 2 is the murderer, or we can say the real suspect of this victims, right? So this is what a simple scene, with the help of DNA fingerprinting, you can able to identify the suspect of the crimes. Clear? Yes, even this DNA fingerprinting is used to prove that Dhanu is a name of the murderer. The human bomb was the real murderer of Sri Rajiv Gandhi. You know Rajiv Gandhi? Yes, former Prime Minister of India, right? Yes. So with the help of DNA fingerprinting, it was estimated that the Dhanu was the real murderer of Rahul Gandhi. I mean to say Rajiv Gandhi, not Rahul Gandhi. It's a Rajiv Gandhi. Okay. So this is the another case. So this is the different applications of DNA fingerprinting. Now there is a short concept regarding the genomics. Now what is the genomics? So it's very simple. Genomics is what? The word genomics has taken root from the terms genome, and you know that genome, chromosome, gene, genome, like that, right? So these are the smallest. Gene is the smallest one, then there is a genome, right? So which is an organism's total genetic constituents is nothing but it's called genome. So genome was first introduced in the year of 1986 by Thomas Roderick to describe the scientific discipline of mapping, sequencing, and analyzing the genomes. Means what are the scientific method to map this particular gene, sequence, and analyzing, right? So it was Thomas Roderick's. Now it is of two types. One is structural genomics and there is a functional genomics. What is structural genomics? Structural genomics is genomics analysis and constructions of high resolution genetic, physical, and transcript map of an organism, right? It is resolution of genetic, physical, and transcript map of an organism. The physical map of an organism developed through the genomic analysis, its complete DNA sequence. That means of structural genomics is a complete DNA sequence. But in case of functional genomics, it is the development and application of experimental approach. It is related to experimental approach is a functional to access the gene function by making the use of information tools provided by the structural genomics. Means whatever the structural information provided by tools, information that is provided, that they are by the structural gene, they're going to be utilized in the functional genomics. So this functional genomics is called the experimental, and the structural one is actually it is a theoretical. Clear? So it is of two types, structural and functional genomics. Now, where this genomics basically use, okay, here there is a slightly mistake, the spelling, that is called genomics. So it is a genomics. Okay, in the previous slide also. So it is genomics. Genomics. Okay, now what is the application of genomics? So genomics is engaged in the identifying the various human gene. Genomics is generally used to identify the various human gene, to give the information about the function of different gene, that what gene is going to perform which kind of functions, that is going to given by this genomics. It is also, it enhance the basic understanding of human genetics. Means if you want to understand the human genetics, this genomics will help you to understand what is actual human genetics. It started from the basics. It give informations that will help to prevent the inherited disease. Inherited means that is called the genetic disorders, like suppose from the grandfather to their offsprings. So such kind of disease, they coming from the generation after generation. So we can easily identify what kind of gene is responsible for this kind of disease. It can lead to the treatment of genetic disorders through the gene therapy. Means with the help of gene therapy, you can treat that gene so that that disease is not going to appear in your offsprings. Right? Another one, genomics has an important role in the animal breedings. So suppose if you want to want a high quality of hybrids animal, then this genomics will help you to get the good quality of good what we can say the version of the particular animals. Means especially it is used in animal breedings. It is useful in the production of transgenic organisms. Transgenic organisms means transgenic plants like BT brinjal, BT cotton. Clear? So these genomics will help you to get the BT brinjal, BT cotton all. So that is possible with the help of genomics. So this is all about the today's session, that is started with the Human Genome Project, DNA fingerprinting and the genomics. So anyone have a doubt in this three concept, because this three concept is the last concept of this chapter. So if you feel that you have a doubt on this, you can ask me. Otherwise we all can move on the question answer session. Yes, any doubt? Okay, I want a response from everyone. Only Kevin and Anne responded. What about others? Okay, Sarjana responded. Okay, so let's move with the next topic. I mean to say the question answer practice. So here you have questions. What is the question? What is it that form the basis of DNA fingerprinting? Yes. What is that form the basis of DNA fingerprinting? I have told you about the VNTRS, and that relate to, I'm not going to tell you the term, but I know that. Oh yes, Sarjana responded very quickly. The relative proportion of purines and pyridines in DNA, no, not possible. The relative difference in the DNA occurrences in the blood, skin and saliva, oh, this is not possible. Then see the relative amount of DNA in the ridges and the grooves of fingerprint, oh no. The another one is satellite DNA occurring as a highly repetitive short DNA. Okay, Yes, I will go with the D. What about your answer? You can also respond. Only Sarjan and responded. What about your answer? I have told you about the repeated DNA short DNA segments or VNTRS that relate to satellite DNA, and it is in use in the DNA fingerprinting. So DNA fingerprinting is based on the satellite DNA, that is VNTRS. Another one, in history of biology, Human Genome Project lead to the development of what? It is related to biotechnology, biomonitoring, bioinformatics, or biosystematics. I think this is very simplest question. Very simple question. And I want answer from everyone. It is very simple question. No one responding. Why? Only Anne and Sarjana responded. What about the others? Prasanti, Kevin, what you all are doing? It's a very simple question. I told you about the computational biology, which is called the bioinformatics. Yes or no? See, Ragul said A, biotechnology. No, we are not related. This biotechnology is not related to Human Genome Project. It is computer related to Human Genome Project, because I told you that Human Genome Project, it was estimated that it will require 3,300 books, and each book is somewhat like 1,000 pages, and on the single page there are 1,000 later, which is impossible to complete wind up within just 30 years. And that's why at that time computer was more flourished. So with the help of computer, they complete this Human Genome Project in within 13 years, and they are using the biological terms in the computer, so it is a bioinformatics. Very simple, but answer is okay. Now next, in the DNA fingerprinting technique, the radioactive intermediate formed during hybridizations are which kind of molecule? It is double stranded or single stranded? This is also simple question. And you know that it is not RNA, it is only DNA. So these two can easily eliminate. Now you have only two options, A or B. Okay, so most of you responded B. Yes, it's a single stranded, but in that answer it is written as a double stranded, but it is a B, which is the right answer. Okay, that is, see the steps which of DNA fingerprinting: extractions of DNA from the cell, then cutting off DNA using enzymes. What is the name of that enzymes? Restrictions endonuclease enzymes. Clear? Then separating with the help of DNA fragment on the gel. Which kind of gel? It is gel electrophoresis, agarose gel electrophoresis. Yes or no? So with the help of, you can separate the fragments and then transferring the DNA onto a paper. What is the name of that paper? It's a nitrocellulose membrane or nylon, yes, on the nylon paper, then adding the radioactive probe, and then setting up the X-ray film, and this X-ray film is called autoradiography. Radiography. Yes or no? Okay. Now the last question. The non-human model organism sequence in Human Genome Project where? Which one is the non-human model organism? It is nematode or fruit fly, wheat or rice, fish or birds, garden pea or fruit fly. Now this is confused for you, right? No? Okay, Kevin, how a nematode is, what is the what is the geological name of nematodes? It is Cinerohabditis elegans. Clear? So nematodes and fruit fly, botanical geological name of fruit fly is Drosophila melanogaster, right? So you can see this is a Cinerohabditis elegans and the Drosophila melanogaster. So these two are the non-human model organisms. This is non-human model organisms whose genomes was sequenced were namely that is Cinerohabditis elegans and the Drosophila melanogaster. Is this clear? Okay, now this is the summary what we studied. We studied about the Human Genome Project, which was launched in the year of 1990 and it was completed in the year of 2003. We also studied about the goals, salient features and the application of Human Genome Project. We also studied about the DNA fingerprinting, which was developed by the Alex Jeffrey, and the process started with the isolation of DNA, digestion of DNA by the restriction endonuclease, separation of DNA fragment by the gel electrophoresis, then followed by transferring of separated DNA fragment by the synthetic membrane. So when transferring it is called the southern blotting technique, and fragment of synthetic membrane on the nitrocellulase membrane. So nitrocellulase membrane, cellulose membrane or nylon is important. Now hybridizations using the labeled VNTR probe. VNTR is what? Variable number tandem repeat sequence. Okay, and detection of the hybridized DNA fragment by the autoradiography, or simply it is called the X-ray film. Clear? This is what the complete process, and we also a little bit discuss about the genomics. So this is what the complete process of DNA, and now I completed this chapter, also the topics, today's topic. Now I request all of you to please move on your whiteboard, I mean to say dashboard, and there is a class assessment with the name of Human Genome Project and DNA fingerprintings. So you all can attend that questions. Yes, please all can move on the dashboard and you can find there is a class assessment already assigned to you, class assessment as well as homework. If it is visible, then start attending. Yes, and done so fast. Are you sure? Okay. Okay, let's do other also. So we will, if you have doubt, then we will discuss. Yes, and any problem? Okay, Kevin, done. Yes, and you want to discuss any question? And can you hear me? Okay, question number 10. Okay, so question number 10 in my system, it is probe using in DNA fingerprinting are initially. Is it you're talking about? Yes? Okay. So what is the problem in that question? You can unmute yourself.
Sir, can you explain why everything includes as an answer?
Okay.
Because with explanation when you gave, I just heard the word meaning right, the others I didn't hear, or maybe I didn't listen properly. So
Okay. See here.
can you explain
Yes.
how everything counts
Yes.
as correct answer?
Yes. See here. Probe. See, probe used in the DNA fingerprinting are initially. So probe is what? It is a single stranded RNA, or it can be a DNA, because it is a probe, right? This probe separately. See in the, I have already listed in over here. Yes, here you can see. See, this is the on the sequence number 6. You can see probe you have to separately create. How you will create the probe? This probe can be the part of, either it can be RNA, or RNA means single stranded RNA, or probe definitely, as I told you, that it is a mini satellite, VNTRS, right? And 19 base long nucleotide. So in the previous day I told you about the different bases of the nucleotide of the probe, like of these satellite. So there are total three satellites we have discussed, right? Three DNA satellite we have discussed. Yes or no? Yes. So in that three satellite, you can see this mini satellite is 19 base pair long nucleotide. That means of all the answer for the probe, because we're not talking about the actual process of fingerprinting. How these probes are prepared? It is from the single stranded RNA, mini satellite, and 19 base pair nucleotide. Clear? Okay. Apart from that, any other question? Sarjana, all question is clear to everyone? Okay. So if all question is clear, then I'm going to wind up this session. And there is a DRS in the coming Saturday. On coming Saturday, we have a DRS on this chapter. So it is my simple request to all of you, please go and revise this chapter again and come up with the full preparation so that we can try to solve maximum questions. As you know that there is a chapter assessment. So yes, chapter assessment is there, but apart from that, if you want to discuss something other or any topic, that we can discuss in that session. Okay, so for today, I'm going to wind up this session. Thank you very much.